GDP attached to it. This ligand binding to the receptor triggers the replacement of the GDP with GTP. Okay, so I've got this receptor that is activated by the ligand that triggers this GDP to be replaced with GTP, okay, which now activates this G protein. So now my G protein is active because it has GTP on it. Okay, and so when that G protein becomes active, it moves over across the membrane. So it moves laterally along the membrane to interact with this enzyme. You'll see how the G protein there fits into that enzyme. So it moves laterally along the membrane to activate that enzyme. And so that enzyme then is what's going to, when that enzyme becomes activated, okay, that enzyme will now activate my pathway. Okay? And all of that gets triggered, again, by that ligand, by that signal molecule. It's this step-by-step process here. Okay? So again, the ligand triggers the G protein linked receptor. Okay? That triggers the replacement of the GDP with the GTP. My G protein then moves laterally into this enzyme to activate the enzyme. We're still all along the plasma membrane here. Okay? And then that active enzyme really triggers my pathway. And my pathway is what's eventually going to cause my response. Okay? And so once that pathway is activated, okay, the whole thing kind of backtracks in reverse. That GTP, just like ADP, okay, it would be a G triphosphate. Okay? So it's going to lose that phosphate. Okay? It will cleave off that phosphate once it has gone over and activated the enzyme, which will then deactivate the whole thing because now it goes back to having GDP instead of GTP. So it would inactivate the whole system. So these G protein receptors, okay, these are used, that's what those yeast used. So those yeast mating factors use a G protein receptors. Okay, epinephrine uses a G protein receptors. If y'all don't remember what epinephrine is, it's adrenaline. Okay, so epinephrine uses it. Some of your hormones will use it. Those neurotransmitters and that local signaling, they use these G protein pathways. Okay, so all of these various things can use the same pathway. Okay, the pathways that get triggered inside of the cell, okay, it triggers a variety of pathways. It will trigger some developmental pathways. Okay, so it'll trigger developmental pathways, sensory perception, in particular for vision and smell. In case you cannot tell, I've totally given up on trying to write nicely on these things. It's just not going to happen. Okay, so the various pathways, again, they can trigger would be developmental ones, sensory perception, vision and smell, bacteria. Okay, bacteria interfere with these, and that's one of the ways they can make us sick. Okay, is our bacteria toxins mess these pathways up. Okay, and so they can cause disease within us. For example, cholera, botulism, as well as pertussis. Some of y'all have maybe heard about pertussis. It's making a comeback in the news. Okay, so all of those diseases come from bacterial toxins interrupting these G protein linked receptors. Okay. All right, so let's look at our next kind of receptor. So our next receptor is what's called a tyrosine kinase receptor. Okay, so a tyrosine kinase receptor. And these are significant because they can activate more than one of those signal transduction pathways that are going to eventually make the cell actually have a response. Okay, so our signal transduction pathway, which we'll talk about when we're done with our receptors, but they will activate more than one of those. So that's your really big difference here between your G protein linked receptors and your tyrosine kinases, is that your tyrosine kinases can activate more than one of these signal transduction pathways. Okay, so when these tyrosine kinase receptors are inactive, they are two separate polypeptide chains. Okay, so I've got two separate polypeptide chains here, and they have, we call them these tyrosine tails that are down in the cytoplasm. Okay, so they're inside of the cell. So with these tyrosine tails, right, those tails have receptors for ligands. Okay, so let's look at what this will look like once they get activated. Okay, so here we can see the inactive receptor over here, okay, versus the activated receptor here. So here's our ligand binding site. So when the ligand binds to the receptor, okay, that will move the polypeptides together. So see, they close this gap here, okay, and they join together into what's called a dimer. Okay, so they are now in the activated form in the dimer. And so that dimer gets phosphorylated. And we need to become very comfortable with this word and this term. Phosphorylation, phosphorylated. Okay, so when we phosphorylate something, we add a phosphate to it. Okay, and so this dimer is phosphorylated by ATP. Remember, ATP has 3 phosphates, ADP has 2. So ATP gave one of its phosphates to these tyrosine molecules. So wherever there's a tyrosine on this dimer, it will receive a phosphate. Okay, and so every single one of these activated tyrosines can trigger a cellular response. So what I have here now is I've got something that can activate multiple pathways. This is important during things like cell reproduction, where I need to activate more than one biological pathway at a time. So like I said before, that's really our main difference between the Gprotein versus the tyrosine kinase. The Gprotein receptor can just activate one pathway, where these tyrosine kinase receptors, because they have multiple tyrosine places that get phosphorylated, they can activate multiple pathways. Okay, so let's look at our third kind of receptor, our ligand-gated channel. So remember, the ligand is just the signal molecule. Okay? So a ligand-gated channel is essentially a gate, a locked gate, that when the ligand binds to it, the gate opens up, and it allows things to travel through it. Okay? So it's going to be one of those integral proteins that spans the entire width of the cell membrane, and so it just opens and closes in response to the ligand. These are very important for ion flow. Okay? For instance, sodium, neurotransmitters in the neuromuscular junction. We showed that picture of the neuromuscular junction. When those neurotransmitters are released from the neuron and bind to the muscle, they trigger an influx of sodium. Okay, calcium channels are controlled by these. So these are, like I said, they're just gates. They are protein channels that are opened up when the ligand binds to them, and they are closed up when that ligand releases. Okay, so let's take a look at, now that we've got the signal has been received. So some kind of receptor has been activated, whether it's a Gprotein, a tyrosine kinase, or a ligand-gated channel. And let's check out now the transduction. So the transduction is our processing of the information. Okay, it's the processing of the signal so that the cell can actually produce the action. And most of our transduction pathways are going to be done by what are called second messengers. Okay, so what do you think the first messenger is? Well, hopefully you told me that the first messenger was the ligand that came in there in the first place. Okay, the hormone, the neurotransmitter, the growth factor, whatever it is. Okay, so this right here would be our first messenger. And so the second messenger is what's going to be inside of the cell that's going to help amplify this transduction pathway. So these second messengers, they're going to amplify the transduction pathway. So they're going to make it have a bigger response and actually have a reaction. And our two most common ones that we're going to talk about here for second messengers are cyclic AMP, okay, or CAMP. So cyclic AMP, as well as calcium ions. Okay, and so we'll talk about both of those. Keep in mind, again, though, when we're talking about these second messenger pathways, if we're referring to a hormone, that's going to have to be with an amino-based hormone. Because remember, the steroid hormones come all the way inside. Okay, so we're going to start with the cyclic AMP. Okay, so here's our cyclic AMP. Cyclic AMP is a derivative of ATP. Okay, so you see here I've got my ATP molecule, and I've got my enzyme here that alters my ATP molecule, okay, and it gives us my cyclic AMP. You notice it got rid of this diphosphate here. Okay, so cyclic AMP is going to act as a second messenger. Let's go through kind of an example of one. Okay, so let's look at epinephrine. What would happen with epinephrine here? Okay, so epinephrine is a hormone. Okay, it's adrenaline. And so epinephrine would be our ligand here. So the epinephrine is going to bind to the Gprotein receptor. Okay, and then remember the Gprotein gets activated. So here's my GTP, comes in, activates my Gprotein. So now it's active. So it comes over and it will activate that enzyme. Remember, the enzyme is inactive until the Gprotein scoots over and turns it on. Well, if you come back up here, you'll see that the adenylate cyclase is what forms the cyclic AMP. So now I've activated the enzyme that makes cyclic AMP. So I am now actually making something inside of my cell that can cause a response. So this enzyme starts converting this ATP, see it fits up there nicely, starts converting that ATP into cyclic AMP. Cyclic AMP gets this pathway going so that the cell will end up finally increasing blood glucose levels. So one of the things that epinephrine does for us is it prepares us for fight or flight. Okay, all those things that happen when we get ready for fight or flight. Our breathing increases, our pupils dilate, our heart rate increases. And one of the other things that happens is that our glucose levels in our blood, in particular shuttling out to our skeletal muscles, will increase because the preparation is that we're going to need more ATP. So we need more glucose to be able to do cellular respiration. So epinephrine is going to raise these glucose levels by triggering this pathway, this cascade pathway. Activates the protein, the Gprotein activates the enzyme makes the cyclic AMP. The cyclic AMP is what actually causes the cell to have a reaction and to release that glucose out into the blood. Okay, so let's look at calcium. So the majority of calcium that is in our cells is actually kept up out of the cytoplasm. Okay, you can see here it's either pumped completely out of the cell. Okay, so it's removed from the cell. This would be a form of, hopefully you saw that, active transport, required ATP. It is a pump. Okay, so it's either pumped out of the cell, okay, it's pumped into the endoplasmic reticulum. Okay, in muscle cells, there's a specialized endoplasmic reticulum that stores calcium because calcium is required for the muscle cell contraction, or it's pumped into the mitochondria. But essentially, you're not going to find calcium hanging out here in the cytosol. So when that happens, that triggers that calcium can act as a second messenger. That will trigger some sort of pathway within the cell. So let's take a look at. Okay, so let's take a look at this cell. So this would be our normal cell up here. Whoa, I don't know why that's so fat. Okay, so this is my normal cell. And this normal cell, like I said before, does not have a lot of calcium out here in the cytoplasm. Okay, so not a lot of calcium there. Calcium is in the endoplasmic reticulum. It's outside of the cell. So what we have here is we've got our first messenger coming in. So there's our ligands coming in. So those ligands have come in and they have triggered the proteins. Okay, they've triggered this Gprotein over here. They've triggered this tyrosine kinase receptor here. Okay, so the Gprotein receptor triggers the formation of DAG. Okay, DAG is diacylglycerol, okay, and inositol triphosphate. Okay, and so I've got this inositol triphosphate, and the IP3 is going to open up these calcium. So I've got every single kind of receptor happening right now in this pathway. I've triggered Gprotein receptors and tyrosine kinase receptors. Without those, I couldn't make the IP3. The IP3 comes and acts as the ligand for my gated channel, and now it releases the calcium out. And that increase in calcium, the calcium will bind with this calmodulin protein. Okay, and that calmodulin protein is what's going to actually trigger my cellular response. Okay, so it's this step-by-step process. Let's talk for a second about muscle contraction. So in muscle contraction, we have to have the release of the calcium for the muscle to be able to actually shorten. If this calcium right here never gets released, then the